Hi class, this is Sir Alex Basco and welcome to our lesson in General Biology 2. Today we are finishing our discussion in lesson 6.2 as we discuss this part 3, which is all about Calvin's cycle. So in part 1 we've discussed the introduction on photosynthesis. On part 2 we've discussed the first part of the stage of photosynthesis, which is light dependent reaction, which in the end we created ATPs and NADPH. In here, what we're going to do is finish the second stage or the last stage of photosynthesis called Calvin cycle, or in other words, the light independent reactions. So let's begin. So Calvin cycle, let's have a little bit of overview what is happening with Calvin cycle. First, it requires CO2 or carbon dioxide, as opposed to light-dependent reactions in the first stage of in photosynthesis, which it requires water or H2O from the roots. In here, in order for us to do Calvin cycle, we require CO2. And CO2, as we know, comes from the aerial portion of the plants, and it diffuses into the leaf by via the stomata of the plant, which is the pores that you will find in your plant. This CO2 then enters the stroma of the chloroplast. If you guys remember what your chloroplast is, this is where the site of photosynthesis is happening. This is where you will find your thylakoid. This is where you will find your stroma, your grana, etc. Stroma is basically the fluid-filled space uh, of the chloroplast, which basically engulfs the whole thylakoids or the grana found inside, that you can find inside in your chloroplast. What that means is that the Calvin cycle is actually happening on the stroma, as opposed to the light dependent reaction. The light dependent reaction is happening on the thylakoid itself, on the grana, but the Calvin cycle is happening on that fluid portion part of your chloroplast, not the thylakoid. Calvin cycle is happening on the stroma. And also here, in order for us to do Calvin cycle, we need the ATP and the NADPH that we have created from the light dependent reactions. And lastly, the ultimate goal of the Calvin cycle is for us to create G3Ps, or the glyceraldehyde 3-phosphate, which then combines ultimately to make glucose. At the beginning of the Calvin cycle, we usually have to have 3 carbon dioxide, and these 3 carbon dioxide create 3 or 6 G3P. There are 3 main stages of Calvin cycle that we are going to talk about today. First is carbon fixation, the second part is reduction, and the third part is the regeneration. So let's begin with carbon fixation. So in Calvin cycle reactions, the first part that we are going to discuss is called the carbon fixation, this part right here. So this whole diagram right here is your whole Calvin cycle, and this part right here is the first part of your Calvin cycle, called the carbon fixation. If you remember what carbon fixation is, this is basically where we fixed carbon. What we mean by that is we take an inorganic carbon, such as the carbon that we find in our CO2, and then attach it or attach it into a molecule that is organic, which in this case, our organic molecule would actually be 3-PGA. So the first thing that would happen is, first we would have to need oxygen or CO2. So the carbon dioxide enters the leaf, passed by the mesophyll system, and into the chloroplast that you will find in your mesophyll cells. And then in the chloroplast's stroma, the whole Calvin cycle starts to happen. We're in an enzyme called ribisco, which is short for RUBP, which is a ribulose biphosphate carboxylase oxygenase. It's quite a mouthful, so just remember the word ribisco. Ribisco is your enzyme the ribulose biphosphate carboxylase oxygenase, which this ribisco, what it does is it catalyzes the CO2 that we have and then attaches it into a carbon, which is what we into a 5 carbon sugar, which is what we call an RUBP, or ribulose biphosphate. So this is the whole process right here. We have our 3 carbons of CO2. Again, we usually, for the whole Calvin cycle, we need 3 carbon, or we need 3 CO2. And then the Rubisco attaches it to our RUBP, creating basically a 6 carbon. So what happens is that the RUBP becomes unstable because from being a 5 carbon sugar, which is one right here, so that's 1, 2, 3, 4, 5. But when the Rubisco attaches the carbon found in the CO2, it becomes 6 carbon. 1, 2, 3, 4, 5, 6. What happens is it becomes unstable and it splits to stabilize itself. And when it splits, we create 2 copies or 2 molecules of 3 carbon compound called the 3-phosphoglycerate, or 3-PGA. So that is the first step of Calvin cycle, or the which is the carbon fixation part. So in summary, we 3 molecules of CO2 enters your plant via the stomata of the leaves, and then the Rubisco takes this carbon dioxide and catalyzes it with your RUBP, or ribulose biphosphate. And then what happens is you create a 6 carbon chain such as this, which are very unstable, which then separates into 2, or in this case 6 3-PGA. 6 because we start with 3 carbon dioxide. If you only have 1 carbon dioxide, then 2 copies of 3-PGA because it splits, right? Okay, so in the first part of carbon, or in the first stage of our Calvin cycle called carbon fixation, we ended up having 6 molecules of 3-PGA. So 1 CO2, 1 carbon dioxide makes 2 3-PGA, which means the whole Calvin cycle usually needs 6 carbon dioxide. So in the end, we make 3 carbon dioxide, which in the end we make 6 3-PGA. So that is the first step. Now let's move to the second step of our Calvin cycle reactions, called the reduction. So what happens? After we create 6 molecules of 3-PGA, what happens is that each of these 6 molecules of 3-PGA, they receive a phosphate group from ATP. So which means we need 6 ATPs. As you can see right here, we have 6 ATPs, and the one of the P's, this is an adenosine triphosphate. 1 ATP contains 3 phosphates, and one of the phosphate group that you will find on your ATP will be donated to your 3-PGA. Okay. So it phosphorylizes the 3-PGA, turning it into a diphosphorylated or biphosphorylated molecule called the 1,3-biphosphoglycerate. Phosphoglycerate. So 3-PGA is just first 3-phosphoglycerate. 3 is actually the number of the carbon. So when we mean 1,3, which means the 1 carbon and 3 carbon contains a phosphate. Biphosphate because we have 2 phosphate, and the phosphate is found on 1 carbon and 3 carbon. So from 3-PGA, which our 3-PGA is just 3-phosphoglycerate, right? Our 3-PGA, it catalyzes a phosphorylated, which we create 1,3-biphosphoglycerate. And then what happens is that we ended up with about 6 ADP as our byproduct. And then the second stage of our reduction is we take this 1,3-biphosphoglycerate and then we reduce them. When in chemistry, when we talk about being reduced, they gain an electron. So if you guys are familiar with Leoger, lose electron reduce, right? Gain electron, sorry, Leoger, lose an electron oxidizes, gain an electron is reduced. So let me write that. This is something that is common in chemistry called the Leo goes Ger, which means when something, a molecule loses an electron, it oxidizes. Lose electron, oxidize. Ger is gain electron, it's reduced. So our 3-biphosphoglycerate is reduced. It means it gains an electron, and the electrons come from NADPH. So the NADPH donates 2 electrons to our 1,3-biphosphoglycerate, and then one of the phosphate group in our 1,3-biphosphoglycerate actually is removed, or it loses one of its phosphate group. So it gains 2 electrons and it loses one of its phosphate, which eventually would lead into the creation of the glyceraldehyde 3 phosphate, or the G3P. So that is the second part of reduction. So the whole process of reduction is, so after the first process of carbon fixation, which we have 3-PGA, the 3-PGA undergoes phosphorylation, wherein it becomes biphosphorylated. The phosphate group is found on the 1 carbon and the 3 carbon. So you have your, this is your 1, 2, 3. So this is your 3 here. This is your 3 PGA, 3 because your phosphate is at the carbon 3. So this is carbon 1, carbon 2, carbon 3. And then when it's phosphorylated, one of the carbon is attached on carbon number 1. That's why you have 1,3-biphosphoglycerate. And then lastly, NADPH donates 2 of its electron, and the 1,3-biphosphoglycerate loses one of its phosphate. So that's one phosphate right there. And then this whole molecule right here now becomes the glyceraldehyde 3 phosphate. Okay? So that is the reduction part of our Calvin cycle. And the last part is what happens to these 3 or G3P is that some of the G3P, more specifically one G3P molecule, actually exit this whole cycle and proceeds into our glucose synthesis. This is the whole another process, but we're not going to talk about this. Just remember that after the reduction process, wherein we create 6 molecules of G3P from our 6 molecules of 3-PGA. One of those G3P goes to make glucose, it exits the cycle, while 5 or most of the G3P are actually recycled into the whole Calvin cycle. And what happens is that these 5 G3P undergo regeneration. We're not going to talk about the whole process, and what happens is that this 5 G3Ps uses ATP to actually recreate the 3 molecules of RUBP, or ribulose biphosphate, that we have lost during the carbon fixation, because we are utilizing this RUBP during our carbon fixation. So there is some need in order for us to recreate this. So they are basically recreated from our 5 or from the 5 molecules of G3P, which we create during reduction. So that is basically what's happening. So in the end, 3 carbon dioxide creates a 3 G3P. 1 G3P is used for glucose synthesis, while the 5 other G3P is recycled to create the RUBP. So G3P uses 3 ATPs to recreate 3 RUBP. So just a little bit of summary in our molecule count. 3 carbon dioxide combines with 3 RUBP, ribulose biphosphates, and makes 6 G3P in the end. So you have 6 carbon dioxide. The Rubisco enzyme catalyzes these carbon dioxides to 3 RUBP, which in turn creates 6 molecules of 3-PGA, and then during the whole reduction process, this 6 3-PGA becomes 6 G3P, right? So in the end, we utilizes 9 ATPs is used to phosphorylate, converting 9 ATPs to 9 ADPs. 6 are used during carbon fixation because or during not only sorry, not carbon fixation, this should be reduction. So 6 ATP is used during reduction, and then 3 during regeneration. And lastly, 6 molecules of NADPH is used during also reduction. Okay, so that's how easy Calvin cycle is. That's how basically the whole process of how the plants create glucose. So that is the whole process of photosynthesis. I hope you guys have, or I hope you guys understand this. If you guys have any question, do not hesitate to message me. But that is all our discussion for today. Thank you so much, and may you guys have a great day. Thank you.